So we're going to be going over a biology overview. And before I get started, I want to talk to you guys about how in biology, chemistry, physics, and math are all included. And biology is the study of life, and some more important facts is in biology, you usually learn about energy, DNA, and cells. So living things that convert energy from one form to another and use this energy to grow, change, or produce is usually in biology. So some questions on this is how does this happen? How does the food we eat allow us to think and do all the things we do? Where does the energy in our food come from? And how did life emerge? How are we similar to a plant or insects? Humans are similar to insects, trees, bacteria, etc. Focus in biology, energy, and the role of energy in life. Important molecules, DNA, its role in reproduction and containing information, cells, evolution, and natural selection. And so this is from Khan Academy. I want to emphasize that. So we're going to be going on the section of what is life. So non-living things might show these characteristics, but it is mostly living things that do. Number 1, organization. They contain specialized coordinated parts. This is what organization means. All living organisms are made up of one or more cells considered fundamental units of life. Unicellular organisms, inside each cell there are molecules which make up cell organelles and structures. Multicellular organisms, humans, these are mostly humans. These are made up of many cells, may be specialized to different jobs and are organized into tissues, connective, epithelial, muscle, and nervous tissue, and these tissues make organs such as heart or lungs. So I drew some pictures. This is the connective tissue, this is the epithelial tissue, this is the muscle tissue, and this is the nervous tissue, and this is a drawing of the unicellular bacterium. 2. Metabolism. It is interlocking chemical reactions allowing organisms to do work, and life depends on it. Living things must use energy and consume nutrients to carry out the chemical reactions that sustain life. Definition: the sum total of biochemical reactions occurring in an organism. Anabolism: the organism made complex molecules from simpler ones and they consume energy. Catabolism is organisms that make simpler molecules from complex ones. They make stored energy available. And these anabolism and catabolism are two types that are subdivided from metabolism, and they are the opposite of each other. Moving on to homeostasis. This is the maintenance of a stable internal environment, meaning the environment inside the body, it doesn't matter of external environment. Example: our human body temperature needs to be at 98.6 degrees Fahrenheit, meaning 37 degrees Celsius. 4. Growth. In the regulated growth of living organisms, including individual cells, become larger in size, and multicellular organisms accumulate, meaning gather and build up, many cells through cell division. Growth depends on muscle building called anabolic pathways that build large complex molecules like proteins and DNA. 5. Reproduction. Living organisms can reproduce to create new organisms. It can be asexual, one-parent organism, or sexual, two parents. This is how a single-cell organisms are. They can reproduce by splitting themselves in two. For sexual, meaning two parents, is called fertilization. Two-parent organisms produce sperm and egg cells that contain half of the genetic information, and these cells form a new individual with full genetic set. 6. Response. Living organisms can show irritability as in response to something that isn't right or is a change. Example: humans pull away their hand fast when they go near flames. 7. Evolution. Genetics of a species changing gradually over time happens to all organisms that are living. Natural selection is included in evolution, and it is where a heritable trait allows an organism to survive and reproduce better at a certain environment. And there is also adaptation. This is when this heritable trait from natural selection becomes more and more common in a population, allowing population to be better suited in an environment. I have to remember that above list of life isn't the exact list that defines life, but it helps us distinguish living and non-living organisms. Question: What does it mean to be alive? This question is not defined yet. Viruses. Viruses don't have cellular structure. It cannot reproduce without a host. It is unsure if they can carry out homeostasis and metabolism. So this is the structure of a virus. Here the green part is the protein coat, and this red is the nucleic acid genome. Remember, viruses aren't defined whether they are living or non-living according to the properties of life listed above. But once again, these aren't the final and the properties of life. Moving on to the scientific method. So a person named Hassan ibn Al-Hassan, he lived in Egypt 1,000 years ago. He was a famous astronomer, physicist, and mathematician, and he was the one who created the scientific method. So in the scientific method, there is a total of 4 steps. So 1, you make an observation. 2, you ask a question, why? 3, try to create a testable explanation, which is called the hypothesis. Example, this is a good example. Salt water has lower freezing point than fresh water. A bad example is that it's a fairy that performs magic on the pond to freeze it faster. Then 4, you design an experiment. In your experiment, you predict. In this example here, there is number 1, fresh water, which is distilled. Distilled means that through evaporation, all the impurities are taken out. Number 2 is salt. We take the distilled water in number 1 and put salt in it. So we can predict that fresh freezes at 0 degrees Celsius, but salt water doesn't. Then we test it. If it is correct, you should just leave it as, or you can try to do additional tests to confirm and revise so it is more specific. If it is incorrect, you should go to the next step, which is refine and iterate. So we come up with a new hypothesis and repeat this step over and over again. Moving on to control experiments. So control experiments are scientific tests done under controlled conditions. One or few factors are changed at a time, others are kept constant. So an example is, we're going to look at teas. So this is the same pot. We put 10 per seed, but one has water every daily, one doesn't do water. And this is experimental, this is control. So the one with water has 9 out of 10 seeds that grew. The one without it had 0 out of 10 seeds that sprouted. So independent variable is the amount of water, and dependent variable is a fraction of seeds that sprout. And I want to emphasize that independent variable is a cause. Its value is dependent from the other. And if I have more different, the factor different between the control and experimental groups. Dependent variable is the effect. The value depends or changes in the independent variable. Measured to see if the treatment had effect. An experimental group receives treatment and experiment. When the control group doesn't, you should have many of experimental group, but control group you should always have at least one. Experimental data is a singular of data is datum. It's observations made during the experiment, and this is it for the notes. And I'll go ahead and post these online.